Hang on. It's a pleasure to see everyone in this way today. Sarah and I are going to give an update on where HCA is now, as well as a forward-looking view towards where we see it progressing in the next couple of years towards the first draft version, and in the fullest of time towards the finalized version. And I will start in our typical tradition. We always alternate this. So I will start this time and Sarah will continue. And so just to remind everyone, we are here all together in order to create comprehensive reference maps of the types and properties of all human cells, the fundamental unit of life, as a basis for understanding, diagnosing, monitoring, and treating in health and disease. And we're going to speak to that today. We fondly sometimes call the HCA a periodic table of our cells, and we try to highlight by this two features. The first is that we really want this complete catalog of all the cells that are out there. But in addition to that, we want to understand the principles in the same way that the periodic table of the elements is not just an aggregation of a lot of elements, but it has some order and structure to it, and it was even able to predict elements that were not yet observed experimentally. HCA kicked off its planning process in October 2016. We are now in August 2021. We went through a lot, including a pandemic that is not over yet. And in this process have done many things, including bringing together this community through many different meetings, conducting the actual work of the Atlas in collecting and analyzing data, and being a good sister initiative to many other initiatives that are focused on the disease states. And we'll speak to all of that today, and we'll start with our current status. And so the HCA community is rapidly growing globally and remains a very open and vibrant initiative. Currently, we have over 2,200 members from more than 1,100 institutes from 77 countries. And one of the things that really excites us is that if you looked at this similar slide a couple of years ago, the representation we saw in many countries in Africa and Latin America was much less substantial than the one we have today. And it's really thanks to leadership in those countries that we're able now to really claim the global nature fully of the HCA, everything except Antarctica. This is also really reflected in the participants in this meeting. Today, we have more than 1,600 attendees. Of course, that's the benefit of the virtual platform. So we're tenfold larger than in a meeting that we would have in person from 59 countries. And in some cases, we have, of course, attendees that are not currently registered members of the HCA. So we strongly encourage you to actually go online and join HCA. The framing for today's conversation, both in this talk and throughout the meeting, is really our roadmap towards a first draft and then a comprehensive atlas. So to remind you, we originally set a rough timeline of 5 years, during which we want to accomplish a first draft with most major tissues, analyzed at least at the histological level, with a cellular and a spatial branch, focused on healthy donors, limited glimpses into disease, of course, from both genders, diverse geographic and ethnic distribution, and some age diversity, with the goal of the comprehensive atlas to encompass all tissues, organs, and systems, have really a deep dive into each organ, into many nooks and crannies, again, with both cellular and spatial information, still focused on the healthy donors, but possibly with additional mini cohorts of disease where we can see unique states of cells, even of healthy cells in the disease context. Again, the genders, but an expanded geographic and ethnic diversity and an expanded age diversity. And I would say that we are actually in pretty good shape on the first draft, as you will see in a moment, but there is a lot of work to be done. And of course, the pandemic itself, the last year and a half, has really changed some things from where we thought that they would be. One of the exciting advances that has happened in the last couple of years is really the maturation of the HCA Biological Networks. Each of them now has clear coordinators, and they span many different organs and systems, all the original 12 ones that we highlighted at the white paper, but additional ones because the community charged ahead faster and with much energy and engagement and with great leadership from many, many scientists across the world. We encourage you not only to join a biological network based on your interest and work with the coordinators to really become well embedded in it, but in the context of this meeting, we have set us biological network booths that are happening today, and visiting those booths can help you understand what the network is about and how to work with it. These networks have been very, very busy, and in aggregate, across 18 different research areas, there are now samples profiled in the HCA from almost 7,000 individuals from over 8,500 samples and spanning nearly 47 million cells, or to be precise, 46,924,322. This is per our latest survey. We run a survey now every quarter when we take a census of all of the efforts in the Atlas, and we're very grateful for all of you for participating. In addition to those cells, there is now a vibrant organoid effort. The network is led by Christoph Bock and Barbara Treutlein, and they have profiled almost 150,000 cells from organoids specifically for HCA. And in addition, during this pandemic, there was a massive effort across all HCA labs to really contribute to the understanding of COVID-19, and we will speak to that today, profiling more than 5.6 million cells in that context. And as you can see, this really covers the many different organs. This is now going to span 2 slides. We can no longer cram it on one. Some systems with particularly high representation. For example, you can see the massive effort in the gastrointestinal tract, including thanks to a really vibrant gut cell atlas led by the Helmsley Charitable Trust. But really in all of the different organs, there is now growing and substantial representation, and that allows new areas such as the cartilage and bone really to come to the fore as well. And here it is for additional types of organs and systems. I will highlight here the massive effort to characterize human development, as well as to increase genetic diversity, which Sarah will speak to a little later. Overall, if you look at it from a quantitative point of view of where we were and where we are now, there was a substantial jump that happened throughout 2019 and the beginning of 2020. Of course, in 2020, given the pandemic, for healthy cells, there was really a pause in the collection effort in most of the labs, as labs mostly shut down for a while, but it is now picking up speed again. And we see it both in the growth in the number of cells, but also in the number of individuals and samples, which suggests that we're really seeing the diversity and the depth that we need in order to know that we capture rare cells and some level of human diversity. Now, overall, this is of course exciting and encouraging for us, but the data are really getting there, not just in adult tissue, but as I said, also in development and more. But you might ask yourself, how close are we to an actual atlas? Well, the data coordination platform has ingested and stored data. And currently there are more than 13.5 million cells worth of data from 64 organ systems and others, 1,200 donors, many projects and many labs. But, and these are centrally located together with multiple types of informative metadata. And of course, the census is much larger than the data in the DCP, and that is for 2 reasons. The first one is that some of the data is not yet ready to be shared. And the second is that some of the data is controlled access, and the DCP is still working on allowing us to share controlled access data and not only pure open access data. But aside from these, there are several very big challenges that remain, and these are especially around the processing of these data, and in particular, using it to construct an atlas that can be queried at multiple levels. So for example, the DCP put together some software pipeline, but we know from our experience that some of the crucial tasks like self-filtering and batch correction, they're not just software problems, and as a result, data in the DCP in its present form requires additional processing before it can be used for downstream analysis. In parallel, we also provide user-contributed matrices, albeit, in different kinds of formats, and these usually reflect the study generator's own work, and that sometimes, as many of you know, involves months of QC and harmonization and a lot of domain knowledge, and so it's sometimes harder to put together. And in Sarah's part, you'll see a little bit how we're tackling these efforts going forward. Now, this user knowledge of technical and batch effects will typically lead to data that's less prone to technical noise than data that's generated just by a single uniform pipeline. So we need to find a way of bringing these 2 views together. And as a result of that, moving forward, we anticipate to have a pivot from how these data are stored and curated, which is the first thing we had to do, towards the development of methods of how they're probed and used. And this will be led by the analysis working group, whose involvement in the initial DCP was limited to date. So this work, which will be led by cutting-edge computational research groups, is going to focus on these data sets, on how these data sets are presented and queried so that we can move from our nice data and data store to a full integrated comprehensive atlas. And in the second part of this talk, Sarah will speak to this more. Now, a second, when we originally highlighted HCA, we said HCA will be a reference for disease. And we truly believed it in the deepest possible sense. We said there were so many things that one could do. With high resolution data, they could change how we do diagnostics of disease. They can identify signatures and new biomarkers as clinicians monitor patients in clinical trials and in the real world. We can use them to identify better reagents for regenerative biology and cell therapy. We can take the data from genome-wide association study and start with disease genes and put them in the right place in the body and follow up on that to understand disease mechanisms and ultimately develop therapies. I think none of us imagined when we originally made this graphic and made this claim that COVID-19 would happen, but it did. And the HCA community has made a substantial effort to try and contribute to the understanding of COVID-19. And we think this really highlights the critical role of these kinds of technologies, approaches, and conceptual advances in understanding disease. So there are 93 HCA publications to date, starting from 2017. 17 of those are COVID-19 related manuscripts, and I'm just flagging here a very small smattering of those. In the first part of the pandemic, we really focused on using the atlas that already existed, the Healthy Atlas, in order to understand COVID-19. We spent a lot of time on this in the previous general meeting, trying to understand the key cell types important for transmission, what might be happening in epidemiology, the pathogenesis of what could happen in infected cell types, and even some guesses into disease mechanisms. This initial effort was led by the Lung Biological Network that really sprung into action in February of 2020, and in an international effort collected data from many, many different labs, integrated it together, studied it, and delivered a lot of important conclusions on all of these aspects, starting from transmission and ending with the model organisms that have served the community really well. But as the pandemic was actually raging in all of our community's attention shifted from the Healthy Atlas to actually collecting COVID-19 cell atlases, both using samples from the blood, looking at patients with mild, moderate, and severe disease, as well as convalescent patients, and analyzing their cellular profiles to understand disease and the nature of neutralizing antibodies, as well as looking at samples that are more invasive from patients, from living patients, aspirates and nasal swabs, and bronchoalveolar lavages, and finally trying to understand the damage that the virus inflicts on the body inside its organs by studying autopsies. And I'll show you a quick smattering of those kinds of answers, of those kinds of results. And so focusing on PBMCs, there are actually, as you saw, there's 5.6 million PBMCs that have been profiled from COVID-19 patients. I'm showing here just one example with about 800,000 cells. This is from a study led by Maz Hanifa in the UK, looking at a coordinated immune response by using the peripheral blood as a window into patients at different levels of severity into disease, and highlighting the roles of a whole set of cells, starting with myeloid cells and CD1, CD16 monocytes, as well as the role of megakaryocyte progenitors and activated platelets, but highlighting also cells such as circulating TF8 cells, clonal CD8 cells, and IgA and IgA2 expressing plasma cells, and those that go up and down in the response, and how this relates to the different conditions of the patients. In parallel to that, there were autopsy efforts. Two of the large ones happened in New York City and in Boston. This was not surprising in the US because these were two of the hardest hit cities in the United States in the early stages of the pandemic. Two efforts in New York City, one highlighted on this slide from Ben Izar in Columbia, New York Presbyterian, and the other led by Chris Mason, I'll get to it in a second, from Cornell Weill. And one integrated effort in the Boston area, spanning all the major hospitals in Boston, Beth Israel, Mass General, and Brigham and Women's, as well as scientists at Harvard, MIT, and the Broad Institute. And they focused on the lung, kidney, liver, and heart. The study from Boston together with data collected also in New York City and collaborated on together in the integration, gave us atlases of the lung, heart, liver, and kidney of patients with COVID-19. And looking at the heart showed the many different ways in which the heart of a COVID-19 deceased individual tried to save itself but failed. And in this process, identified cell states that we have seen before in conditions such as idiopathic pulmonary fibrosis, like PATS, a way in which one type of alveolar cell is trying to become another type of alveolar cell, as well as cell states that were not seen before in human, like IPBLPs, which you can think of as the emergency reservoir to try to replenish the 81 gas exchanging cells in the body, as well as other features of fibrosis. This allowed us to suggest a model of multiple failed paths for alveolar cell regeneration in severe COVID-19, including not only the ones that exist in the healthy lung of 82 self-renewal and 82 to 81 cell differentiation, but also additional ones that are these emergency paths that unfortunately in these patients have failed. In the study in New York City, they have dug further into the lung autopsies at their disposal, find not just a large diversity of cell types and differences in cell proportions amongst COVID-19 and control lungs, and the same impaired lung regeneration, which gave all of us confidence that this is a general finding, but also different kinds of aberrant immune responses, and suggested a model where some of the failure of 81 cell regeneration is related to signals that come from the impaired immune response and kind of put the regeneration on the wrong path. And together, these kinds of studies have allowed us to show that we can learn rich things from a disease atlas, even from a small number of patients, but we need a reference atlas in order to do that. The reference atlas allowed us to annotate the cell types, allowed us to compare the cell compositions, allowed us to know which cell states are healthy and which cell states are aberrant, as well as allowed us to clarify which cells are being infected by viruses by reading viral reads into annotated cell states. We also, and this is the effort from Chris Mason's lab in New York City, some of these studies also looked at the lung in the spatial context because viral infection is not uniform, and looked at regions with high and low viral load, as well as could compare them to samples from flu and ARDS. This is with the spatial transcriptomics profiling method from NanoString in this particular case. And this allowed identifying not just the differences between patients, but also the differences between different infected regions and the programs that are active in those regions. Overall, all of this data is available in open resources for everyone to peruse. Much of it was released far before publication so that scientists could immediately use the data as soon as it was appropriately processed and annotated. And there are many different portals and mechanisms for visualization and access to both raw and processed data now for the community to pursue their studies in COVID-19. And with this, I will pause and let Sarah switch. I have given you a general overview of where HCA is now and its status, as well as its contributions for understanding disease. And really the rest of our pivot now is to think about consolidating all of this into an integrated cross-tissue atlas. And Sarah is going to focus on that in the second part of the talk. I'm going to stop sharing and Sarah will share her slides.
Fantastic, thank you, Aviv. And I will indeed share. Can everybody see this?
Yes.
Excellent. And as Aviv said, I want to now think forwards about how we get from a draft to a comprehensive atlas. And for the first version of HCA, of the HCA Atlas, in the white paper, as you heard, we put forward that there would be extensive single-cell RNA sequencing data, and we now also have nucleus profiling and at the RNA and attack epigenetic level, as well as multiomics. Spatial approaches are gathering apace, and for many tissues and organs, there's gaining level of coverage also at the spatial level in terms of tissue sections using diverse technologies that you can see here, both at the protein and RNA level. And there are continuously newly emerging approaches that allow us to calculate, to characterize tissues in space. And I want to give a shout out here to the wonderful Standards and Technology Working Group that's done a lot of work on discussing, disseminating, comparing existing methods, and providing gold standard benchmarks to help the community in terms of the comparative strengths and weaknesses of different methods. And one example of this work is from the Space TX team, benchmarking spatial transcriptomics, and providing a common computational framework for comparing the different methods in terms of also useful for data analysis and visualization of the data. Here, I want to highlight a bunch of different ongoing benchmarking approaches for cell and nuclei technologies that you can see here carried out by many different centers. And also the Kidney Biological Network for activities in this area in terms of both mouse and human kidney, and comparing data in different, using different modalities, and that's summarized in the paper below. Aviv has already talked about the data coordination platform, and obviously having the data uploaded and queryable in the data coordination platform is really important for an HCA version 1.0, and it's great that we're getting more and more studies into this one common place for people to go and query and download the data, and we'll talk more about that in the next session. The analysis working group has been doing an amazing job, and of course the co-chairs are Donna Pear and John Marioni, and there are 8 fabulous members that are part of this working group that does incredibly important work that's absolutely at the heart of the human cell atlas community in terms of organizing meetings, jamborees, focusing on techniques for data processing, integration, analysis, and also the cell annotation platform that I'll come on to in a minute. Of course, for a human cell atlas version 1.0, what we need is for the individual tissues and tissues within individual organs to be covered as thoroughly as possible in terms of annotating and assembling the data. And again, the Lung Biological Network has been a sort of front runner. Of course, they're also kind of the oldest biological network in the history of the HCA by a few weeks or so, thanks to the pandemic and the pressure to basically rapidly organize ourselves, particularly with respect to the airways. And you can see here work from Malte and Fabian's group, also 2 lung biological network coordinators, Sasha Mishar and Martina Wayne, on benchmarking atlas level data integration, single cell genomics, using multiple lung data sets as an example. And this is really where all the biological networks are going to be heading going forward, is taking the data and integrating it for a single organ. And the cell annotation platform that Peter and collaborators are developing, and that we'll hear about later in this meeting, is an enabler of this kind of activity, because it provides a sort of marketplace for encouraging convergence on consensus nomenclature of individual clusters, cells, and so on. And the initial release is planned very soon, so watch this space. Of course, given all that data and the integration, we need to, it's incredibly useful for the broad biomedical community, not just the kind of computational aficionados, to be able to query the data in a user-friendly manner. And there are different axes for this, including not just the cell types, but also gene expression programs, individual genes, obviously, ultimately relative positions of cells and co-occurrence in terms of their physical locations and tissues. And we look forward to the discussion right after this session about this. So there are various axes that are that we're moving towards completing a draft 1.0 human cell atlas. And so it's a really exciting juncture and a really exciting time in the history of the project in this sense right now. I'm going to spend the last part of the talk just looking forwards very briefly before we finish towards the HCA version 2.0 and more comprehensive completion. And what's very useful for this are efforts that not just focus on a single tissue or organ, like, I highlighted the lung just now, but efforts that basically go right across organs and tissues and provide kind of an integrated view of an atlas of the body, albeit usually at lower coverage than the individual focuses on organs. And one example is, of course, the work from Guo and collaborators published last year in Nature that you can see here for the human cell landscape. Another one that we know is coming up soon and that's on this portal is Tabula Sapiens from the West Coast, looking at 8 donors and 25 organs. And then we have other kinds of studies, and one example is here that I'm a coordinator and collaborator on, that Cecilia Dominguez-Conde from my group has put up on bioRxiv together with our collaborators, where we're studying immunity across 15 tissues in the human body, both lymphoid and non-lymphoid. And what we can learn from that is the systemic nature of the immune system and the sharing of clones, for instance, across nearby tissues in the body. Also lessons on particular isotype usage in different parts of the body, in the B cell compartment, and other new insights into immune system across the body. So these systems like immunity, vasculature, and so on go across the body and can also provide a kind of framework of the whole human cell atlas. Equally, there's another cross-tissue atlas coming up that I'd like to highlight, which is using GTEx tissue samples. And this is from Aviv's group and collaborators, and is in preparation using nuclear RNA sequencing from frozen tissue, also giving insights into the myeloid compartment across tissues with the M1 and 2 skewed macrophages in different compartments, and an interesting glycolipid metabolizing macrophage present in scored across tissues, present across all tissues, more prevalent than some in others. And the other interesting thing that I'd like to highlight in this study is this sort of scaffold nature, providing one data set characterized with a uniform method, and then using that to learn lessons from other data sets that you can see down here from published data by scoring against it. And so this is the kind of approach that we'll be using going forwards to assemble a deeper atlas in terms of version 2.0. The other axis that we are obviously actively embarking on as a community is capturing genetic diversity. And the 1K project from Joe Powell and collaborators is an example of this, capturing 1,000 blood samples from 1,000 individuals, albeit at a lower, at a lowish coverage of 1,000 cells each, which then allows single cell eQTL mapping to identify cell type specific expression variation, finding 27,000 eQTLs that are cell type specific, and also providing insights into autoimmunity. And another example is work by Jimmy Ye and Mina Subramanian and colleagues showing cell types where frequencies, state, or a combination of the two change in systemic lupus erythematosus, looking at also a largish number of donors, 136 patients with SLE58 healthy controls, 1 million cells sequenced across them, coupled with in vitro characterization of changes. And the final axis that we want to go towards for version 2.0 is, of course, different stages of life. And this includes from embryonic to fetal to pediatric stages of life. And what I'd like to highlight here is what I'll call a roadmap perspective, where the Human Cell Atlas Developmental Biology Network PIs got together and put forward a kind of status report of where the human developmental biology community stands, and where do we want to go going forwards in terms of sampling different embryonic periods, using different technologies, spatial, RNA-seq, in situ sequencing, and so on, to assemble together maps of developing organs and indeed embryonic and fetal stages, and learn about what coordinates organ development across the whole embryo. I also want to highlight that you may think, why do you want to understand human development? There are very clear translational applications. So just like Aviv highlighted for the human cell atlas as a whole, there are many different applications for learning about disease. We've learned a lot about COVID-19. Even human development, plays a crucial role in informing us about engineering cells and tissues in vitro. In other words, regenerative medicine. Engineering tissues, also therapeutic targets in both pediatric and adult diseases. Related to the axis of human diversity and genetics that I alluded to before, I want to give a big shout out here to the work of the Equity Working Group, and that's co-chaired by Musa Mlanga, Parth Majumdar, and Alex Shalek. They've done a stellar job together with the other 14 members in promoting and supporting the progress towards equity in the human cell atlas. And that means engaging the global community, empowering them to participate in human cell atlas, but also providing regular diversity audits in terms of not just the membership, but also the collected data. In other words, the samples that we're studying, working with funders to encourage them to earmark funds to increase recruitment of scientists and study of samples from across the world, so that we have an inclusive human cell atlas and an inclusive human cell atlas community. And the funders have been fantastic partners in this. Related to this, but separate, I'd also like to give a big shout out to the Ethics Working Group. This is co-chaired by Bartha Knoppers, Barbara Wald, and Roderick Gigo, and consists of 16 members and 10 observers. And it's there to discuss the ethical and legal issues relevant to HCA. And on this, I'd like to encourage you to go to the General Data Protection Regulation talk by Sarah Bowers, which is also related to this working group. And it's really important to understand these issues, not just for human cell atlas, but for the whole genomics community. Finally, I'd like to emphasize that the human cell atlas is a partnership with funders that support us and consortia that are funded to study the healthy human cell atlas, but also very close relationship, of course, with the human disease communities, and they're shown over here on the right-hand side. I just want to emphasize that it's really a fantastic partnership across both of them, and we're making great progress towards our goal. And with that, I will end here, say thank you very much, and perhaps there's time for questions now.
I believe, John, that there were a couple of questions. Do you want me to read them out loud? I guess so. So the first question it was from Tiago Lubiana. Is there so far an estimate of how big the periodic table will be? How many reference cell types and states are expected? So that's the issue with the analogies. They start well and then they start breaking quickly. I'll say two things. I think we have learned two things in the process: that categorizing cells into cell types is an extremely productive endeavor up to a point. And at some point, it stops being the correct way to describe what we're seeing. And so I think it was in one of Sarah's slides on the querying, one component of what we want to do is really find those neat categories that come with a clean label on them, put annotations in a cell annotation platform, have signatures that describe them, and continue. And I think we're still seeing completely new cell types that fall into this category, but they mostly tend to fall into rare cell types that you start seeing only when you have a very large number of cells that are sampled or a very unique region that is being sampled. But what makes it a little harder to give a full answer to your question is that we also see a magnificent amount of cell states and programs, which are not, especially programs, meaning different combinations of genes that go together, and you can sometimes find the same program in multiple different cell types. I highlighted some examples in the COVID-19 where the type of cell is not really a category there for some of these things. It's really a program that might characterize either a transient state for a cell or just a particular program inside a cell that otherwise is a fixed entity. And those are a different axis for us to describe what we're actually seeing, and there could be a substantially larger number than them. For me, the most appealing thing about talking about periodic tables is hoping that we would understand something about the principles so that we would be able to move to predict either the number of expected cells or the presence of a particular kind of cell that we haven't observed yet from, we could use evolutionary conservation for that, or maybe we would understand the regulatory code better. I mean, we learned our lesson from the Human Genome Project. We're not trying to bet on the number of cells, because anyone who tried to bet on the number of genes in the human genome turned out to be wrong. So we're trying not to go that route, but really get the data.
So I would encourage also, can I give a shout out to the ontologies talk this afternoon? Please attend that. That's in the session later, and by David Osumi-Sutherland. It's about naming and categorizing cell types and cell states, and that could be illuminating on this topic. Just to say that it's probably much, the numbers, however, whatever word you use, are much bigger now that we've got HCA going than they were before. So there's a huge number of discoveries that have come out of human cell atlas studies regarding cell types and cell states.
And, a shout out both to the ontology talk that will happen in conversations, and also it's everyone's collective responsibility to participate in the annotation efforts, because that relies on domain expertise to really tease apart things and give them names. And until they have names, people have really a hard time working with things in biology. Sarah, I think I'll turn the second question to you. How do the spatial approaches work? 2 minutes about all spatial genomics approaches.
So I went really quickly, and I encourage you to attend some of the talks.
We have a whole session
in this meeting on this topic, and also other topics. But in a nutshell, basically, the spatial approaches characterize tissue sections, which can be thin sections of 10 microns or so thick, and also thicker sections, let's say, even up to a centimeter cubed. And so instead of getting information on suspension cells or nuclei, where you're looking at one at a time, you're gathering information on the cells in their native tissue context in 2 or even 3 dimensions. And the methods can be sequencing based, so direct readout of the transcriptomes at different positions by next generation sequencing, or they can be microscopy based using probes either for the fluorescent probes for the RNA, fluorescent probes for the protein, that are read out in a variety of different ways, either combinatorially or individually. And I think I'll leave it there because it's a really huge topic, very exciting topic, an evolving landscape of methods development. Of course, you can appreciate that to make a human cell atlas, these are going to be absolutely crucial, and the insights that we gain from them are just amazingly exciting in terms of understanding histological structures in human tissues. And I think that we can squeeze in the last question. Aviv, I'll hand it back to you. Any interest to go to the other extreme of aging, I think referring to how I talked about development, i.e. the elderly? Yeah,
that's actually a tough one. First of all, yes, and there are efforts already to try and characterize aging. Aging is tough, because what happens with aging is that you have a gap between chronological aging, just the age of the person, I'm so and so years old, and biological aging, physiological aging, which is how old, in a sense, is their body and their cells. And those two things start becoming decoupled, which is why some people actually appear to age faster in some ways than others, and have more age-related diseases than others. That also gets convolved with all those age-related diseases. Having said that, I think there is a huge interest in this, as well as the hope that, in fact, looking at cells at different adult ages could help define better clocks for physiological aging, a real phenotype by which to define that. I would not say that this is fully within the scope of HCA to wrestle aging too into the ground, but that it is similar to the overlaps that we have with disease atlases, where we do a portion of the effort is HCA related, but a full-fledged answer in any given disease, maybe COVID-19 being an unusual exception because of the circumstances, but a full-fledged study in any given disease becomes really the focus of a directed study. So that is, I think, a partial answer to, again, a very big question. I think there are additional remaining questions, but we would have to answer them online, given simply that the time is up for this session, I'm told. But Chrissy Kim Lim, for example, has just asked a question and we'll address it by text inside the chat.
Yeah.
I believe we're taking there's going to be a 2-minute pause in the switch to the panel.
That is correct. We will take a quick 2-minute break and we'll see you back here shortly. Thank you, everyone.
Thank you.